Have you ever wondered how scientists amplify a single piece of DNA into millions of copies in just a few hours? The secret lies in a powerful technique known as the polymerase chain reaction, or PCR. PCR, first developed in the 1980s by Kary Mullis, remains a cornerstone of molecular biology, genetics, and forensic science. But how does this fascinating process work? Imagine you're a detective and you've just found a single strand of DNA at a crime scene. You need more of this DNA to analyze it, to identify the culprit. This is where PCR comes into play. Firstly, we need to heat the DNA. Why, you ask? To separate the two strands of DNA. This process, known as denaturation, occurs at a high temperature of around 95 degrees Celsius. Next, we move to the annealing step. As the temperature decreases to around 55 degrees Celsius, short pieces of DNA called primers bind to the separated DNA strands. These primers serve as starting points for a new DNA strand. Following this, we increase the temperature to around 72 degrees Celsius for the extension phase. Here, an enzyme called Taq polymerase attaches to the primers and adds nucleotides to extend the DNA strand. This results in 2 new copies of our original DNA. And there you have it, the 3 steps of PCR: denaturation, annealing, and extension. By repeating these steps around 30 times, we can create millions of copies of the DNA in just a few hours. But why is PCR so important? Well, it has numerous applications. It's used in genetic testing, identifying diseases, forensics, and even in research to study ancient DNA. So, to summarize, polymerase chain reaction is a powerful technique that allows us to replicate a single piece of DNA into millions of copies. It works in 3 main steps: denaturation, where the DNA strands separate; annealing, where primers bind to the separated DNA strands; and extension, where the DNA is replicated. This technique has wide-ranging applications from disease identification to crime scene investigation. In the vast world of biology, PCR stands as a testament to our ability to understand and manipulate the very fabric of life. It's a small process with a massive impact, and its discovery has truly revolutionized science.